Okay, and so let's take a look at your general tests for your proteins. Okay, and so under this, you have there your Ninhydrin test and your Biuret test. Okay, and so you have there your Ninhydrin test and your Biuret test. Okay, before that one, and so let's take a look first at your proteins, okay? And so your proteins, and so they are actually a diverse group of macromolecules, okay? And so they are organic compounds of high molecular weight, okay? And so they have a high molecular weight. Comparing it to your carbohydrates, these proteins have a relatively higher molecular weight than your carbohydrates, okay? And so with this one, they are a group of diverse molecules, and this diversity is directly related to the combinatorial possibilities of your 20 amino acid monomers, no? And so you have there your different amino acids. You have probably your 20 different amino acids, okay? And so the combinations of which, of course, will form your protein. And that is the combination also is what you call this responsible for the diversity or the complexity of these proteins, okay? And so they are made up of alpha amino acids joined by peptide bonds, okay? And so they are joined by peptide bonds, and then the total possible number of combinations for such a molecule is enormous. And so you have actually more, no? You can reach up to trillions of combinations. But then, and so few of these combinations are produced by living organisms. Okay, and so that is for your proteins. Take a look at your what you call this diversity of proteins accordingly is demonstrated by the complex set of functional and structural properties of your proteins. And so first on the list, and so you have your structural features that make protein folding is relatively rapid and a successful process. And then the presence of your binding sites that are specific for one or small group of molecules, and then an appropriate balance of structural flexibility and rigidity so that the function is maintained, and then your surface structure that is appropriate for a protein's immediate environment, and then of course the vulnerability of proteins to degradation reactions when they become damaged or no longer useful. Okay, and so you have there. Moving on, your proteins accordingly are distinguished based on, and so they are distinguished based on their number of amino acids, and so or your amino acids present actually in your proteins, they are called as your amino acid residues. They are called as your amino acid residues due to the because in the formation of your peptide bonds, and so when you join amino acid, one amino acid and another amino acid, you have what they call the reaction is it involves a dehydration reaction. That is why, and so the amino acids present in your peptides or in your proteins, they are called as amino acid residues. Okay, and so you have there, and so actually it depends on the number of amino acids present, and so the overall amino acid composition in their amino acid sequence. Okay, and so you have there, and so your molecules with a molecular weight that ranges from several thousands to several million daltons are called as polypeptides, no? And so your peptides, they are what you call this generally low molecular weight, and they are what you call this your polypeptides, and so they are made up of 40 to 50 amino acids, while your proteins, and so they are made up of more than 50 amino acids in the sequence. Okay, and so they consist of your proteins consist of one or more polypeptide chains. Okay, and so again, your term protein here, and so is given to a molecules with uh, more than 50 amino acids in the chain. Okay, and so that is moving on. You have here your illustration of what you call this, an illustration of your proteins, no? And so you have here your myoglobin, and so this one is your collagen, and so a structural protein, your myoglobin, and so these are found in your muscles, your lysozyme, for what you call this digestion of your microorganisms, Okay, you have your purine, your cytochrome C, your hemoglobin, and so this one is responsible for the oxygen carrying capacity of your blood. And then your deoxyribonuclease, this one is an enzyme. This one is an enzyme also, your catalase. And so those are examples of proteins, no? Okay. Let's move on to your amino acids. Okay, and so your amino acids, and so they are produced from the hydrolysis of your polypeptide, no? And so when you hydrolyze your polypeptides, and so they produce your amino acids. Okay, and so your amino acids, they are amphoteric. Okay, and so they are amphoteric, and so they can behave as acid or base, probably. Okay, and then they are Zwitter ions also, and so they are Zwitter ions, no? And so they bear positive and negative charges. Okay, and so they are amphoteric. And then they are amphoteric, of course, that is for uh, your carboxyl group accordingly at pH 7, and so your carboxyl group of an amino acid is its conjugate base form, no? And the amino group is the conjugate acid, okay? And so thus, and so if you say conjugate acid, it's not really an acid, no? And so, and your amino acids, they are biologically active, and so they are biologically active, and so in a way, and so you have here your several, what you call this, your several alpha amino acids or their derivatives accordingly act as chemical messengers. And so, for example, again, and so you have your glycine, your glutamate, your gamma aminobutyric acid. Your GABA here is a derivative of your glutamate. And then your serotonin and your melatonin, these are actually derivatives of tryptophan. And these ones, this glycine, this glutamate, this GABA, your serotonin and melatonin, they are chemical messengers or they are neurotransmitters. Okay, and so they are neurotransmitters. Okay, your thyroid hormone thyroxine is actually a derivative of tyrosine. Okay, and so your thyroid hormone thyroxine is a derivative of tyrosine, and this one is a hormone. Okay, and so this one is a hormone. Okay, and then, of course, they what you call this are precursors of your amino acids are precursors of nitrogen-containing molecules, no? And so you have the example, of course, you have your nitrogenous base components of your nucleotides and your nucleic acids. And then finally, and so they act as metabolic intermediates. And so they act as metabolic intermediates. And so recall, and so when we talk about your uh, urea cycle, and so they are your urea cycle and your Krebs cycle, and so you have there your involvement of your amino acids, like you have your arginine, your citrulline, and your ornithine. Okay, and so again, your urea cycle, that is the disposal pathway of excess nitrogen. Okay, and so moving on. And so you have here your peptide bond formation. And so again, and so when you join together your amino acid and the two amino acids, they form a peptide bonds. And so when you add more and more, then you form peptide bonds. Okay. And so your peptide bonds, they are amide linkages formed when the unshared electron pair of your alpha amino nitrogen atom of one amino acid attacks the alpha carboxyl carbon of another in what we call this a nucleophilic acyl substitution reaction, no? And so again, your peptide bond formation, and so that is what we call this. Your peptide bonds are amide linkages, and so formed when your unshared electron pair of your alpha amino nitrogen attacks the alpha carboxyl carbon of another in what we call this in a nucleophilic acyl substitution reaction. Okay, and so this one, and so is actually a dehydration reaction. Okay, and so this one is a dehydration reaction. That is why when an amino acid is dehydrated, and so that is why, as I have said earlier, an when you join them together, ayan, so you have already, you call them at your as your when they already form a peptide bond, then they are already amino acid residues. Okay, and so you call them as amino acid residues because a water molecule is removed. Okay, and so that is for that. And so your in the formation of your peptide bond, and so there is a removal of water. Okay, and so that is why you call them as your amino acid residues. Again, your amino acid residues, and so they are linked amino acid in a polypeptide chain. Okay, and so moving on. This one actually is a bond formation. And so accordingly, there is a bond. This is an illustration of your peptide bond formation. And so your peptide bond forms again when your alpha carboxyl group of one amino acid reacts with the alpha amino group of with the nitrogen of your alpha amino group of another. Okay, and then of course water is formed here. Okay, and so that is an illustration of your peptide bond formation. Okay, and then you have your dipeptide. And so your dipeptide, and so that is a two amino acids linked together. And so with this, of course, when you have a dipeptide bonds, you have two amino acids and one peptide bond. Okay, and so when you have a dipeptide, you have two amino acids that are linked together. And so you already call them as amino acid residues. And then you have one peptide bond. Okay, and so of course, as the chain lengthens, elongates, and so the prefix denotes the number of residues. So, for example, of course, and so the prefix reflects the number of residues. And so, with that example, of course, you have a dipeptide, a tripeptide, you have a three amino acid residues, a tetrapeptide, you have four, and then a pentapeptide, and then so on and so forth. No, and so, of course, your peptides, and so they are less structurally complex than protein molecules. And so they are less structurally complex than your protein molecules. So, but then you have your peptides, they have significant biological activities. Okay. You have here your what you call this biologically important peptides. Okay, and so you have your tripeptide, and so you have your tripeptide glutathione here, your gamma glutamyl L-cysteine glycine, and that is for your glutathione, and so it contains an unusual gamma amide bond. You have your oxytocin, and so this one, your glutathione, this one is a what you call this, and so it functions as a protective agent for your blood cells, no? And so, and then you have here your oxytocin. This one is a hormone. And so this your oxytocin is a hormone, and so involved in lactation. Your vasopressin. And so this one is your vasopressin is involved in water balance, no? And so, and then of course you have your uh, atrial natriuretic factor, no? Ayan, so your ANP, your atrial natriuretic peptide, together with your brain natriuretic peptide, okay? And so they also, what they call this, for electrolyte balance also, and so those items, okay? And so moving on, let's take a look at your protein functions. And so you already know this one. We talked this about in the lecture. We talked this in the lecture. But then, and so, ulitin ulit natin. Okay, and so you have your protein functions still. And so you have your different proteins with you. And so let's take a look at this. Your what you call this? Your protein functions for structure. Okay, and so we have actually a lot of proteins that functions for structure. And so you have here they provide support for your uh, they provide support. Okay, and so let's take a look at this. And so your structural proteins like your collagen and elastin. And so you have there your collagen, that is a major component of your connective tissue, together with your elastin. And so your elastin, that is a rubber-like protein that is found in your elastic fibers and that is found in your elastic fibers. These elastic fibers are found in your blood vessels and the skin. And so to what they call this, to provide the elasticity for this parts of the body, no? And then you have here your fibroin also is a what they call this, a protein. And so this one is from your fibroin is a silkworm protein, and so it has actually a significant mechanical strength also. And then your protein, the function for movement, and so you have here your cytoskeletal proteins like actin and tubulin here. And so again, your proteins are involved in movements of your cell. And so you have your cytoskeletal proteins like your actin, your tubulin, and other associated proteins. And so uh, are under this function of protein. And then you have your defense. And so they provide protection against microorganisms, against invasion. Okay, and so you have your defense, and so many proteins have a protective function. Okay, and so in for us, for humans, and so you have the keratin, and so your keratin is a protein that is found in the skin, and so your keratin is also a structural protein, no? And so, but then in this context, your keratin, and so this is found in your skin, and so in your skin cells that aids in protecting the organism against mechanical and chemical injury, no? Other than that, you have your immunoglobulins, your antibodies that are produced by your plasma cells, and then you have your of course, these antibodies are produced to protect against invasion by foreign organisms like your bacteria, of course. And then you have your acute phase proteins. Other than those, you have your clotting proteins, you have your fibrinogen and your thrombin. These are proteins that are activated in case there is a tissue injury, and so to prevent blood loss when your blood vessels are damaged. And then you have a lot more under this what they call this defense, no, under your protein that functions for defense. Okay. And then you have your catalysis. And so your catalytic proteins, called as your enzymes, accelerate thousands of biochemical reactions. And so examples, you have your trypsin, your pepsin, you have those items, your amylase, your alkaline phos, acid phos, those items. And then you have your proteins function for regulation. And so your regulation, you have your what they call this, they regulate the metabolic processes. And no, with this, and so this function of your protein is attributed to your hormones. You have your hormones here, and so they what they call this, regulate your metabolic processes. And then you have your proteins also function for transport. And so they function for transport. Okay, and so uh they transport substances across the cell membrane, or from cell to cell, or from organ to another organ. Okay, and so you have many proteins functions as carriers of molecules or ions across your membranes, or between your cells, or in between organs, no? And so you have there. And so let's say, for example, you have the target organ for this hormone, and so that is actually being carried. No, and so you have here examples of this. You have your hemoglobin, and so it transports oxygen. Your sodium potassium ATPase, your glucose transporter, and so your others, your albumin. This one is also a what do you call this? A transporter. Your lipoproteins also, they transport also other substances. Okay, and so those items for that. Moving on, you have your proteins can also function for storage, and so they are reservoir for essential nutrients, no? And so example, your of albumin in birds and eggs, and casein in your mammalian milk are actually rich sources of organic nitrogen during the development of this. And so other than that, you have your from your what they call these plants, like your uh from your germinating seeds, no? And then of course you have also a lot in you inside the body, and so you have your ferritin, and so those items. Then you have here for immune response. Okay, and so for stress response, your proteins also function for stress response, no? And so some proteins can convert variety of toxic materials to less toxic ones, no? And so you have there, and so you have actually your cytochrome P450, and so it actually is a group of diverse enzymes that usually convert toxic organic contaminants into less toxic ones. And so your metallothionein is also an example. And so you have there. And then, of course, your toxins. And so again, and so many organisms produce toxins. No? And so this one is for defense or what they call this in predation in case of that. And so your neurotoxin that are found in your snakes, in the venom of your snakes. Also, the apitoxin from your bee sting. And and so other than that, of course, the toxins produced by your spider, no? And so the spider, your scorpions, those items, your centipedes, and so also. And so you have therefore your functions of your protein. Okay, moving on, let's take a look at your protein structure. And so first on the list, of course, you have your primary structure, no? And so the primary structure, and so that is again your specific amino acid sequence. And so of course with that, you have there the presence of your peptide bonds. With this, no folding, no beta-plated sheet on this one, and so just the sequence. And then you have your secondary structure, and so that is you have already here the folding of your peptide chains. And so your folding here is due to the bonding of your carbonyl group of one peptide chain with the amino group of one peptide chain. Okay, and so you have here your alpha helix. You have here to form of core yours in your secondary structure. You have actually here your alpha helix and your beta pleated sheet. Okay, and so again, in your secondary structure of your polypeptides, you have your several repeating patterns. Okay, and then this one, of course, you have there the folding. Okay, and so you have your alpha helix and your beta pleated sheet on your secondary structure. Okay, and so this one's our example of your beta pleated sheet. This one, your beta pleated sheet and uh your alpha helix. And so you have your alpha helix and your beta pleated sheet. Okay. And then you have your tertiary structure. Okay. And so under this, there is a unique three-dimensional conformations that takes place. Okay. And so you have here your helical arrangement is interrupted with that to allow numerous bonds and additional bonding. Okay, and so you have there. And so with this, of course, you have there your three-dimensional structures, three-dimensional conformations. Okay, and so this one, your tertiary structure is maintained by your covalent bonds. And of course, you have here your polypeptide chain is bent or folded to form a compact, tightly folded protein structure, no? And so under this, your tertiary structure, and so you have your fibrous and your globular. Okay, you have your fibrous and your globular. So by the way, and so in your three-dimensional shape conformation, your conformation here is you have a combined two secondary and tertiary structure of the peptide chain. And so again, in your conformation, you have a combined secondary and tertiary structure of the peptide chain. Okay, and so under this, you have your fibrous and globular proteins. And so your fibrous, you have your fibrous one, and so you have there your collagen, that is a fibrous protein. And then your globular, and so you have your globular, you have your hemoglobin, that is a globular protein. Okay, and so this one is this one actually illustrates the interactions that maintain your tertiary structure. And so the interactions here are actually described, are actually discussed in the other video. Okay, and so you have here, you have your hydrophobic interactions, you have your disulfide bridges, And your salt bridges here, you have your hydrogen bonds, hydrogen bond formation, and the rest of the interactions. And so, of course, these ones are important to what you call this, maintain your tertiary structure. And then, of course, for your quaternary, and so that is a fitting together separate coiled and folded structures to form a functional one, no? And so you fit, and so you fit individuals, and so you can have a small, many, and so you have there you have your subunits. You have actually in your quaternary, you have what you call as subunits here, no? And so you fit together your separate coiled, they are your subunits, and so to form a functional structure. Okay, and so examples of your quaternary structure, uh, proteins that possess quaternary structure, you have your isoenzymes and your hemoglobin. And so in your hemoglobin, that is actually made up of what they call this, four. And so that is made up of four uh, subunits. And so also for your isoenzymes, and so let's say, for example, isoenzymes of your lactate dehydrogenase, you have four. And so for LD1, LD2, and so those items. Okay, and so that is for your quaternary structure. Okay, moving on, let's take a look at your Ninhydrin test. And so your Ninhydrin test, and so that is your the reagent that you use here is your Ninhydrin in ethanol or acetone. Okay, and so you have the Ninhydrin, and so what we used in the laboratory is your Ninhydrin and acetone. Okay, and so you have here your amino acids undergo deamination and decarboxylation to form an aldehyde. Okay, and so your reaction of amino acid with ninhydrin will lead to the carboxylation and deamination. Okay, and of course there is a formation of your carbon dioxide and ammonia here. Okay, and so with that, and so your ninhydrin undergoes a reduction, and so to form your hydrin. And so hydrin dantin. Okay, and so that is your ninhydrin is reduced to form your hydrin dantin. And this hydrin dantin complexes with ammonia to produce a blue color or violet. And so that is your romanza purple, no? And so this one, and so your blue color or violet, or you have your violet blue condensed product, that is your romanza purple, no? And so all proteins and peptides and amino acids will give a positive result for this. And so all your proteins, your peptides, and amino acids will give a positive result to your ninhydrin test. But then, and so your proline and your hydroxyproline will give a negative result with this one. Okay, and so they will give a negative result with this. Okay, and so other than that, your ammonium salts, your amino sugars, and ammonia will also give positive ninhydrin test, no? And so this one, of course, in order to obtain a reliable results for this one, and so the test solution should not contain the compounds that are mentioned, your ammonium salts. And so to make sure that they are only proteins, then of course you should remove the others, your ammonium salts, your amino sugars, and okay, this one can be qualitative and quantitative accordingly. And so this test, your ninhydrin test, and so is a qualitative and quantitative. Okay, and so and your ninhydrin test, for your information, this one is actually used in forensics, no? And so this one is used in forensics. Okay, and so moving on, let's take a look at your Biuret test. And so this one, your Biuret test, and so the reagent that is used here is your copper sulfate and sodium hydroxide. And so, of course, you have your copper sulfate and sodium hydroxide giving you a blue color of the reagent, no? And so your copper ions bind to the nitrogen present in the peptide bonds. And so it binds to the nitrogen present in the peptide bonds, giving you a rose pink to violet to purple color. And so if you have a rose pink, your violet or purple color, then they are positive with your Biuret test. Okay, and so with this, of course, and so you have it is for your peptide bonds, the presence of peptide bonds. But then your dipeptides will not give a positive result with your Biuret test. your dipeptides again, and so will not give a positive result for Biuret test. And so because the requirement for your Biuret test is you have at least more than, and so you have more than two accordingly. And so because in your dipeptides you only have one peptide bond, then it will give a negative result for that. Okay, and so this one is your Biuret test accordingly is a qualitative, and this one is used to check the extent of protein hydrolysis. And so this one, your qualitative test, and so this one is used to check the extent of protein hydrolysis. And so of course in hydrolysis, and so if mapapansin, if there is what you call this a negative result with your Biuret test, then probably you already reach your the end of your hydrolysis. And so you produced your dipeptides, or you already produced individual amino acids na. Okay. And so in testing for Biuret test, and so that is a what you call this in testing for Biuret test, and so from a Biuret test, and so you use a solution from a salting out process, and so and you used ammonium sulfate, and so accordingly, excess alkali should be added. Okay, and so that is for your Biuret test. Of course, the positive result is your rose pink to violet to purple. Okay, and so this one, and so a table that of the uh, what you call this, a table of the test, I mean the reagents, the sample that we used. And so with this on your Ninhydrin test and the Biuret test, okay? And so for your Ninhydrin test, and so your blue purple or your Rumans purple, and so as the positive result in your Biuret test, and so you have a violet one. And so for your egg albumin, your milk, your tryptophan, your tyrosine, your methionine, and your arginine will give a positive test for it will give a positive Ninhydrin test. While for your Biuret test, and so your egg albumin and your milk will only give a positive test for Biuret. And then your tryptophan, your tyrosine, your methionine, and your arginine, they are negative. And so they are negative. Kasi nga, they are only amino acids. And so this one, your tryptophan, your tyrosine, your methionine, and arginine, they are uh, amino acids. Of course, there is no peptide bonds present in them. And so that is why they will give a negative result to your Biuret, no? And then your egg albumin and your milk, these are your proteins that are actually tested. Okay. And so that ends. And so that is for your general test for your proteins.